on His Majesty's service. Taste receptors. The sense of taste is responsible for the evaluation of food and drink. It serves to detect nutritionally relevant or harmful compounds in food, which leads to acceptance or rejection of potential food sources. Taste sensors are represented by receptor epithelial cells localized in the taste buds of the tongue in fungiform, foliate, and velate papillae, as well as scattered throughout the epithelium of the soft palate, posterior pharyngeal wall, and larynx. The overall quantity of the taste buds can reach 10,000. The taste buds have an ellipsoidal shape, occupy the entire thickness of the epithelial layer, on the surface of which they open with taste pores. Each taste bud consists of 50 to 100 closely adjacent elongated gustatory epithelial cells with apical microvilli extending through the taste pore to contact chemicals in the oral cavity. Gustatory epitheliocytes are divided into 4 types, characterized by both morphological and molecular features. Type 1 cells are glial-like supporting cells sharing many features with astrocytes. They are most abundant, comprising approximately half of the total number of cells in a taste bud. They have electron-dense nuclei and wing-like cytoplasmic processes that ensheath other cells. Type 2 cells and type 3 cells serve as the transducers for different taste qualities. Type 4 cells are small oval cells that lie on the basal lamina. They function as stem cells responsible for 3 other cell types renewal with a life cycle of about 10 days. The sense of taste in humans detects a wide variety of chemicals that constitute 5 main or primary qualities or modalities: sweet, salty, bitter, sour, and umami. In Japanese, umami means savory. Note that the function of the taste system depends on olfaction, texture, and the internal state of the organism. In humans, taste perceptions are modulated by hunger or satiety, expectations, and emotions. Each type 2 or 3 gustatory cell is most responsible to single taste quality, and each taste bud usually contains several cells which can respond to each of the taste qualities. Thus, gustatory cells are chemically tuned, whereas taste buds are not, and there is no so-called map of chemosensitivity on the tongue. It's a misconception that sweet is perceived at the tip of the tongue, bitter on the back of the tongue, and so on. But in the different regions of the tongue, there exist variations in thresholds to different tastes. Due to this, some regions are the most sensitive. Type 2 cells house 2 classes of the G-protein-coupled receptors in the membrane of their microvilli. T1 receptors detect sweet and umami tastes. T2 receptors, bitter taste. 3 different T1 receptors were identified, namely T1R1, T1R2, and T1R3. These receptors function as heterodimers. T1R1 plus T1R3 as the umami receptor, and T1R2 plus T1R3 as the sweet sensor. The umami receptor responds to all L-amino acids in rodents, but only to L-glutamate in humans. The family of the bitter taste T2 receptors consists of about 30 members. Some of them are narrowly tuned, others respond to many bitter compounds. T1 and T2 receptors activate similar transduction cascades in different subsets of type 2 cells. Tostans binding to receptors lead to activation of the G protein termed gastducin. It is similar to transducin of photoreceptors, and as transducin consists of 3 subunits. Gastducin activates phospholipase C, which converts the membrane phosphatidylinositol biphosphate into the second messenger, inositol triphosphate. The latter binds to its receptor, causing the release of calcium ions from the smooth endoplasmic reticulum. This activates a monovalent selective cation channel, and this leads to the opening of voltage-gated sodium channels, depolarization and release of ATP through voltage-gated ion channels. ATP activates P2X receptors on afferent nerve fibers. Thus, type 2 gustatory cells lack voltage-gated calcium channels and other presynaptic specializations, although they associate closely with afferent nerve fibers and use ATP as the transmitter. Type 3 cells detect sour or acidic stimuli. Transduction of sour involves the permeation of protons through ion channels located in the microvilli membrane. The entering protons depolarize the taste cells and block potassium channels, amplifying the depolarization. This activates voltage-gated sodium channels and leads to calcium influx via voltage-gated calcium channels, triggering the release of transmitters. Type 3 gustatory cells secrete 5-hydroxytryptamine, gamma-aminobutyric acid, and norepinephrine. 5-hydroxytryptamine acts on neural receptors located on the afferent nerve fibers. It is inferred that gamma-aminobutyric acid and norepinephrine modulate afferent responses. Sodium and chloride are essential nutrients and must be regularly consumed to replace excreted sodium chloride. That is why this mineral is readily consumed by animals and humans below isotonic concentrations. By contrast, high concentrations of sodium chloride are aversive, which reflects a protective mechanism against hypernatremia and dehydration. The study of salt taste detection in rodents led to the conclusion that it might be divided into 2 components based on the sensitivity of responses to the amiloride, which is epithelial sodium channels blocker. It decreased the responses to sodium chloride in afferent nerve fibers and reduced the behavioral response to sodium chloride solutions when low concentrations of sodium chloride were used. but it was ineffective in high concentrations of sodium chloride. Thus, there are two pathways. Amiloride-sensitive pathway, which is sodium selective for low salt detection, with epithelial sodium channels identified as the receptors for mediating low salt attraction. And amiloride-insensitive pathway, which is cation non-selective for high salt detection. It may be provided by the beta-responsive type 2 cells and/or the sour-responsive type 3 cells. Furthermore, it is suggested that the salt detection mechanism may involve not sodium, but chloride ions. In addition to the above, it should be noted that compared with rodents, in humans who enjoy low salt and avoid high salt, amiloride does not significantly change the perception of the saltiness of sodium chloride. Thus, in humans, the salt detection and transduction mechanisms may differ from those in rodents. Which gustatory cells detect salty taste and what is the transduction mechanism remains unclear. However, in recent experiments on mice, it was shown that in fungiform papillae, a pool of gustatory cells that co-express epithelial sodium channels and voltage-gated ion channels is not large. Thus, the mechanism of amiloride-sensitive salty taste transduction may include sodium entry into receptor cells through epithelial sodium channels, which leads to the opening of voltage-gated sodium channels and depolarization. In response to depolarization, voltage-gated ion channels open and release ATP on P2X receptors of afferent nerve fibers. Besides T1 and T2 receptors, some other G-protein-coupled receptors have been identified in taste buds. They contribute to the detection of nutrients even though they do not appear to mediate a distinct taste quality. GPR 120 and CD36 were shown to detect long-chain fatty acids in the taste buds. Detection of long-chain fatty acids and transduction mechanism are also connected with intracellular calcium ions increase, which results in the secretion of ATP or glucagon-like peptide 1 onto afferent nerve fibers. Thus, the perception of fat has traditionally been considered to rely mainly on food texture and olfaction, but recent studies show that the taste may also play a role in the detection of long-chain fatty acids. However, the qualification of fat detection by taste buds as the main taste modality in humans has remained a subject to debate. In recent years, it has been shown that the T1 and T2 G-protein-coupled receptors are expressed in the diverse organs of respiratory, digestive, urinary, and reproductive systems. They are termed extraoral taste receptors. They do not mediate taste per se, as they are not linked to neuronal perceptive pathways, but they play an important chemosensory role. For example, the gastrointestinal tract is a sensory organ responding to a variety of signals from its lumen, such as nutrient and non-nutrient chemicals, mechanical factors, microorganisms, drugs, and toxins. This sensing plays an important role in the control of multiple functions, including digestion and absorption, motility, regulation of food intake, insulin secretion, and metabolism, protection from ingested harmful drugs and toxins. Detection of intraluminal content is provided in much the same way as detection of taste by the tongue via activation of specific G protein-coupled receptors, which are located on the so-called open enteroendocrine cells, cells with microvilli extending to the lumen. G protein-coupled receptors respond to amino acids, peptides, fatty acids, sugars, artificial sweeteners, and bile acids. Their activation leads to intracellular calcium ions increase, as it was described above regarding taste buds. This in turn results in the release of hormones, for example, glucagon-like peptide 1 derived from L cells or cholecystokinin from I cells. Hormones act via paracrine pathways or enter the circulation or interact with nearby afferent nerve terminals from vagal, spinal, and myenteric neurons. Thank you for attention.